All right, we're recording. So what we want to do in this second part of today's topic is talk about some of the other contributions to digestion and absorption. And that's going to come from the liver, the gallbladder, and the pancreas. And in the diagram we have in front of us right now, we can see the liver. And we can see that the liver is arranged in two distinct lobes here. Just to orientate yourself, that's the stomach entering into the small intestine. And this is the duodenum part of the small intestine. There's our pancreas. And underneath, tucked underneath the big right-hand lobe of the liver is our gallbladder. Okay, now the first thing I want you to notice is that when we look at the when we look at these three distinct organs slash glands, they're going to have their own respective responsibilities and contributions to the digestive system. And one thing we know, or we're going to learn about the liver cells are called hepatocytes. And hepatocytes are responsible for a lot of different types of responsibilities. They have a lot of different functions. But one thing they do is they produce, and almost all of them do this, is they produce something called bile. We mentioned bile earlier today. So bile is going to be drained by bile ducts. What we have is these ducts here would be referred to as the left hepatic ducts. Hepato, by the way, hepatocytes, has to do with the liver. So these are the left hepatic ducts. That would make these ones right hepatic ducts. So they're collecting bile and draining them towards the common bile duct. So here we have the common bile duct. And all of that, the common bile duct is going to drain towards the duodenum. Remember, we said that bile was going to be received by the duodenum along with pancreatic juices and the chyme from the stomach. So here we would have our pyloric sphincter right in this region, and that's where our chyme would come in from. And here we have the common bile duct that's going to drain bile. Let's put our bile in a nice bright green color. So we're bringing bile in from the right and left lobes of the liver and delivering it through this duct system into the small intestine. So when the small intestine is exposed to food, there's going to be a couple of hormones released that stimulate the gallbladder to contract, and the gallbladder stores bile. So we can see that the gallbladder is partially obstructed here in this view by the liver. But the idea here is that bile that was stored in the gallbladder is going to be delivered. So this duct that comes from the gallbladder and joins in with the common bile duct, that duct is called a cystic duct. And cyst is a sac, a fancy word for a sac, and the sac in this case is the gallbladder. So that's where that terminology comes from. That's a cystic duct. And when the cystic duct joins with the common hepatic duct, we create the common bile duct. So this duct here, all a bunch of names, common bile duct. That's the name for the portion of this duct system that is the combination of what's coming from the gallbladder and what's coming from the liver. So we'll talk a little bit about the gallbladder specifically in a couple of minutes. This just represents the pathway that bile is going to be introduced to the small intestine. So if we're between meals and the duodenum is empty, then basically a sphincter down at this entry point here is blocked and the bile is backed up. And it backs up and enters into the gallbladder. So the gallbladder stores bile. It does not produce bile. That's an important distinction. Can almost guarantee you that there's going to be a question somewhere, at least in the multiple choice, that asks you about the purpose or the responsibilities of the gallbladder. So it's basically a muscular sac that stores bile and can concentrate it a little bit. And when food is introduced to the duodenum, a hormone gets released that signals the gallbladder to contract. Gallbladder contracts and a volume of bile, it can be introduced to the small intestine on exposure to food. And bile is particularly good at mechanically digesting lipids. So when we talk about, well, let's finish our picture here. So the other part of this duct that empties into the small intestine is the pancreatic duct. So this pancreatic duct is going to drain from the bulk of the cells in the pancreas. The bulk of the cells in the pancreas, probably in the vicinity of 80 to 90% of all the cells in the pancreas, have a digestive function, and they produce enzymes. And we'll talk about some of those enzymes in a minute when we look at the pancreas specifically. And they produce bicarbonate. And by now we know the bicarbonate as a base is designed to neutralize the acids that are going to come from the stomach. We're also going to find out that the enzymes that are being released from the pancreas are inactive until they're exposed to an alkaline environment, which is what's going to happen inside the duodenum. So we're going to drain, let's make it a different color. We're going to drain from the products of most of the cells in the pancreas pancreatic secretions into the small intestine. So we said when we talked about the duodenum, we mentioned that the duodenum receives chyme from the stomach and bile from the gallbladder and liver, and now pancreatic juices from the pancreas. So that's what we're describing in this picture. And that's where the contributions from these three organs are going to come from with respect to digestion and then absorption. Okay, so we see this picture, we see these three organs that are going to sometimes described as accessory organs to the digestive mechanism, but we can see where they're oriented from a physical point of view. All right, so let's take a look and talk a little bit about our pancreas initially. So the pancreas, and we've talked a little bit about the pancreas before when we talked about the endocrine system, but most of the pancreas is exocrine, which means that it's going to be a gland tissue that promotes a product or produces a product and delivers it to a duct system. Endocrine glands produce a substance, a hormone, and introduce it to the circulatory system in the absence of a duct. But an exocrine gland utilizes a duct. So it's mostly about being an exocrine gland, producing pancreatic secretions, sometimes called juice. And if you're more comfortable with juice, that works. And a smaller percentage, in that sort of 5 to 10% range, is endocrine. And that was our beta cells, our alpha cells, and our delta cells. Beta cells produced insulin. Alpha cells produced glucagon. And our delta cells produced somatostatin. So those are the clusters of cells. If our pancreas looked a little bit like this with a head and a tail, and it had a duct system, like we saw in that other picture, that drains the products from most of the cells in the body and drains them into the duodenum. Within small clusters interspersed between all the regular cells, we have these little islets. A guy named Langerhans was the first to sort of identify these as cells that had different functions throughout the pancreas. And so they used to be called islets of Langerhans, and they're more accurately referred to as pancreatic islets now, because that at least gives us a location when we're looking at a name. And that's the location for these beta cells, alpha cells, and delta cells. They're going to be found in these little clusters. But all the rest of this gland is responsible for producing pancreatic juices. And pancreatic juices are alkaline, so their pH is greater than 7.4 in the body. The secretions are in the vicinity of 1 to 2 liters per day. By the way, I didn't mention the secretions that are produced in the stomach. That's more like 2 to 3 liters per day. And in the small intestine is about 1 liter per day. So the idea there is just recognizing that the digestive system is responsible for the production of a lot of fluid and has to absorb that fluid in order to maintain homeostasis. So a relatively small amount of fluid is lost through the stool on a daily basis, and the rest of this fluid is being absorbed between stomach, intestine, both the small and the large intestine. So just keep that in the back of your mind too. The secretions have to be reabsorbed if we're going to maintain homeostasis of water balance. And you can appreciate that in conditions like diarrhea, we're concerned with dehydration. With vomiting, we're concerned with dehydration because a lot of that fluid that's being lost through those two mechanisms isn't being obviously reabsorbed. It's not being retained by the body. So there's going to be an imbalance created there. So this pancreatic juice is alkaline. It's got obviously water and electrolytes, like all of our water solutions are going to have. And then it's going to have enzymes. And these enzymes have names like pancreatic amylase. So that's working on what type of organic compound?
Carbohydrates.
That's right. Carbohydrates. So remember, we used that word amylase when we talked about saliva. We said that it produced a salivary amylase, and we mentioned then that amylo tells you it's going to work on carbohydrates. So we also have the production of, these are really variations, slight variations, pancreatic lipases, clearly working on lipids, but other enzymes that are going to be released have the names like trypsinogen and chymotrypsinogen. And we know, like we mentioned earlier today, if they end in OGEN like that, they're inactive. So they're going to be released from the pancreas inactive, but when they mix with the chyme from the stomach in the duodenum, they become active. So when they become active, trypsinogen is going to become trypsin, and chymotrypsinogen is going to become chymotrypsin. We've got some carboxy peptidases. And we can appreciate maybe by then with that name, peptidase and trypsin and chymotrypsin, these are going to work on proteins. These are going to be responsible for breaking down proteins to their amino acids. So if they've left the stomach as small chain peptides, These enzymes located now in the small intestine, but coming from the pancreas, are going to finish the job and get them down to their amino acids. Once we get carbohydrates down to monosaccharides and lipids to fatty acids and proteins to amino acids, they're small enough to be absorbed in the small intestine. They're small enough to be absorbed, and they're going to take advantage of every transport mechanism you can think of. Active transport, primary, secondary, active transport, passive diffusion, facilitated diffusion. We're even going to see exocytosis. I mentioned that that's one of the mechanisms that's going to be utilized to deliver chylomicrons from the absorptive cell into the lacteal. So when you think about absorption, think about all of those membrane transport mechanisms that we talked about way back in the first semester there. They all come into play here to help us with the ability to deliver nutrients from the lumen of the digestive tract into the circulatory system. So the pancreas is going to deliver these inactive enzymes that become active in the small intestine, and then we know what happens when we get into the small intestine. So we've got these additional enzymes now being contributed to by the pancreas. Okay, now I got a picture of the pancreas too, just isolated. Well, not that one. Here, this gives us a little bit of a picture of the pancreas. So here we see the pancreas. It's that yellowish structure in the middle. You see how it's related to the duodenum, how close it is to the duodenum, receiving bile from the common bile duct. This would be our cystic duct. This would be the common hepatic duct up here, draining from the kidney, from the liver, and draining from the gallbladder in this case, joins together with the pancreatic duct to enter into the duodenum. The little blue dots represent the islets that represent alpha, beta, and some delta cells. And then we've got these acinar cells, the ones that are responsible for producing the digestive enzymes and the bicarbonate that's going to help neutralize the contents from the stomach. And this also gives you an idea where the spleen is located, just from an anatomical point of view. The stomach would be coming into this area here, partially covering up that spleen and partially covering up the pancreas, and we would have the stomach slightly to the left and the liver slightly to the right. We'll talk more about liver location in just a minute. Okay, let's get this layer. All right, now let's just look quickly at the gallbladder then. Remember, we said we're going to make it look green because that's kind of the color of bile. So it's this smooth muscular sac that has a duct that leaves it and joins up with the common hepatic duct to form the common bile duct. So we got bile coming from the liver and bile that would be coming from the gallbladder. So that's why this is referred to as common bile duct. Joins up with the pancreatic duct to enter into the duodenum. So we mentioned that bile is composed of bile salts. It's water plus bile salts. The bile salts are produced by those hepatocytes, and they emulsify, that's the key word, emulsify fat. And that's a fancy word that means they agitate. The salts will literally agitate, come in contact with fat molecules, agitate them, and break them up. So it breaks up large fat molecules to smaller ones. So that means we're going to take this large fat molecule and we're going to agitate it with bile salts. And that's going to result in the large molecule breaking up into smaller molecules. Now, when we break those up into smaller molecules, we're going to increase. So the result of emulsification is that we increase the surface area of fats so that we have a larger target for lipases. Okay, so think about that for a second. What we're saying is that emulsification of fat is a mechanical form of digestion. Salts are agitating a globule and making it into smaller ones. But when we make it into smaller ones, we create more surface area. There's now more surfaces for enzymes to act on. So this is a great example of a cooperation where a mechanical digestion is going to increase the efficiency of chemical digestion. Everybody understand that?
Yeah.
Right? Because you know I like asking questions about efficiency and what makes something better than another. And here's a great example of that. In the case of the cooperation between bile and enzymes that in this case are being released mostly from the pancreas, but also locally from the small intestine. So now we have more targets and more surface area for these enzymes to act on to complete the digestive process. So once we get the lipases, in addition to the emulsification, we hopefully are going to end up with some fatty acids. And those fatty acids then can be absorbed in the jejunum, and we can then deliver those to the circulatory system and do something with them, make something from them. So that's what we're thinking about with the gallbladder. Now, there's a hormone, and we're going to talk about the hormones at the very end of today. But just so we get some of the, there's a hormone called cholecystokinin. A kinin is a general term for something that encourages an action or a function. In this case, cholecystokinin is, you can see cholesterol in that word, and cysto is referring to this sac. In this case, it's the gallbladder. So it's abbreviated as CCK, and it stimulates the contraction of the gallbladder, amongst some other things that we'll talk about later. But that's why it's called cholecystokinin. So when food enters the small intestine, cells in the small intestine produce CCK. CCK targets the gallbladder and causes it to contract, and that's going to release bile into the small intestine to help with the digestion of fats. Make sense?
Yes.
Okay, so that's a little bit about our gallbladder. Keep in mind the gallbladder stores bile, does not produce bile. Now, let's take a look at this liver. This liver requires a little bit of extra time because it's responsible for a lot of things. So we'll see that not only is it big, but it's got cells that have multiple responsibilities. So the liver is the second largest organ in the body. What's the largest one?
Skin.
That's right, skin. So again, not a question I'm going to ask you, but if your board exams are going to ask you anything, they're going to ask you sort of stupid questions like that. What's the second largest organ in the body? And that's the closest thing you're going to get to an anatomical question, probably on your national board exams. They don't go into too much detail on anatomy and physiology. There are some times when they do, but it's usually a surprise. So again, just something to keep in the back of your mind, not something to be memorizing or anything about that. And it's generally arranged like we showed you in that first picture into two distinct lobes. From a functional point of view, these lobes are filled with hepatocytes. And it's got a connective tissue. The good news is these cells can undergo mitosis, so they can be replaced. It makes them a good organ for donating, because you can donate part of your liver, and that part that you donated will regenerate and replace itself. So you can donate some of your liver and get it back again. That's a nice thing about the liver as far as that goes. But it has a lot of responsibilities. So if the liver becomes unhealthy, for example, chronically exposed to toxins like alcohol, then it starts to lose some of those functions. And that loss of function after chronic exposure to toxins like alcohol is irreversible. So it basically is scar tissue that replaces the healthy tissue and results in some loss of function. That's otherwise described as cirrhosis of the liver. Think of that as a little bit of an oversimplification, but it's basically hardening of the liver. And that's when scar tissue replaces the healthy tissue. So the first consideration when we look at the organization of this liver is the cells. And hepatocytes are the primary cell type that we're going to find in the liver, and they perform all kinds of metabolic, secretory, and endocrine functions. So they're responsible, and we're going to talk about these a little bit later, metabolic, secretory, and endocrine. And they're arranged in clusters that are sometimes described as hepatic follicles, or clusters of cells that are basically going to be formed around some capillaries. And we'll talk about those capillaries in just a second. The next feature that we want to be aware of with respect to the liver is that there are a series of small ducts. We showed you those earlier. Small ducts between the hepatocytes that collect bile. And we know that those ducts are going to drain into larger ducts. The right hepatic duct and left hepatic duct drain into the common hepatoduct, and that drains into the common bile duct, and that drains into the pancreatic duct that empties into the duodenum. We did all that stuff on one of those previous slides. So we're already generally aware of the collection of bile coming from these hepatocytes from all over the liver. But the feature that we want to be a little bit more aware of is what are called hepatic sinusoids. Hepatic sinusoids we know that sinusoidal capillaries, from when we talked about the circulatory system, were the leakiest of those capillaries. So these are very, very permeable, in other words, leaky capillaries that are surrounded by rows of hepatocytes. So surrounded by rows of these hepatocytes. So what we kind of see here is in segments, if we take a segment out of the liver, we would see this central vein, and it's a vein, that's important. And coming off the vein are these sinusoidal capillaries. And clustered around these capillaries are the hepatocytes. Rows of hepatocytes. They run the length of these leaky capillaries. And so, if they're leaky capillaries, we're going to want to see bulk movement. We're going to want to see diffusion from the capillaries into the tissues beside those capillaries, in other words, the hepatocytes. So we're going to find out one of the most important features of this liver is that the liver receives oxygen-rich blood from hepatic arteries. So that shouldn't be a big stretch for anybody. If we got the word hepatic in there and artery, that's going to be part of the systemic circulation. The hepatic arteries are going to deliver oxygen to the hepatocytes so that they get their oxygen in order to be able to make ATP. And they'll get some glucose in order to make ATP and so on. But here's the thing that makes the liver very unique. It also receives nutrient rich blood from the hepatic portal, that's an important word here, vein. So it's a vein, okay? That vein is going to be represented here with this central venule, let's say, that spreads into these leaky venous capillaries, but rich in nutrients. So rich in nutrients, what have I got? That's not what I want. Rich in nutrients is because it's drained the circulation from the digestive system. Remember, we had that villus? We had a villus that had an arterial system. First, we had our lymphatic lacteal, and then we had an arterial system, and we had an exchange going on there, and then we had a vein. And I made the point of saying that the nutrients are going to be delivered to the vein side of this capillary network. Now, if we go back and look at the esophagus entering the stomach, and then entering the small intestine where most of the nutrient absorption is going to occur, okay, and to a lesser degree, we'll put in our large intestine here. I don't want to block up our whole drawing. Okay, but what we want to understand is coming off of the abdominal aorta, we're going to have this trunk. It happens to be called the celiac trunk, but the celiac trunk gives off a branch to the stomach. It's called the gastric branch that fits for the stomach. It's also going to give us the aorta here is going to also give us a branch that supplies the liver. I'm going to put it over here because we're going to draw the liver over here in a minute. So let's put our liver in just so that we've got it as to be able to complete our picture here. So this would be our hepatic artery. Remember, we said it was oxygen rich, hepatic artery. That's our gastric artery. Okay, and coming off this branch, so this celiac branch is also going to have a branch that goes to the spleen, splenic, and then a little bit down below, we're going to have a couple of other trunks that come off. One of those is called a superior mesenteric trunk, and the other one's an inferior mesenteric trunk. And all I need you to remember about that, so this whole thing is an abdominal aorta. Okay, and the names, really the names at this point are not big important factors here. I'm giving you them to be complete. So off our superior mesenteric, we've got the branches that are going to supply most of the small intestine. And we're going we're also going to be able to supply the pancreas and then this inferior mesenteric supplies the colon, ascending, descending, and transverse parts of the colon. Then down below here, we would split off into iliac arteries that go to the lower limbs. So the idea, though, that I'm showing you is here's our oxygen-rich blood. And coming off of the oxygen-rich blood, we're going to drain into veins that leave the stomach and leave, in particular, what we're interested in is what's leaving the small intestine. So they're going to drain into a common vein that, and this is the important part of all of this, this is the hepatic portal vein. And the hepatic portal vein has got all the nutrients that were absorbed from the small intestine. And we can see that it's going to go first to the liver. And the significance of this, okay, this is important, in my opinion. It's important, so probably for you it has to be important. The significance of this is that liver receives the nutrient-rich and that's sometimes referred to as first pass metabolism. In other words, the nutrients that are being absorbed from the small intestine go to the liver first. And that means the liver gets to use them first. So that's going to be part of the metabolic responsibilities of these hepatocytes. So when they get the first exposure to carbohydrates, monosaccharides, glucose especially, they're going to be able to determine, they are able to determine whether the first responsibility should be to restore blood glucose levels. In that case, they're going to release the glucose right away. If blood glucose levels are good, they're going to convert the glucose to glycogen. If glycogen levels are maxed out, they convert the rest of the glucose to triglycerides, and the triglycerides will be delivered to adipose tissue and stored there. That's what we're talking about when we talk about first pass. When the fatty acids are delivered to the liver, the same concept's going to apply. They're going to take the fatty acids, and if, for example, we need cholesterol for every cell membrane in the body, for example, then the hepatocytes will start to synthesize some cholesterol and then take some of that lipid and create carriers for those lipids in the form of lipid proteins. They're called lipoproteins, low density lipoproteins that are abbreviated as LDLs. They deliver cholesterol to every cell and tissue in the body. And HDLs take extra cholesterol that's in the body, in the periphery, and bring it back to the liver. When they bring it back to the liver, it can either be cholesterol can be released and degraded there, or released and re-released out into the circulation as LDLs, depending on the requirements, depending on the needs. But HDLs are sometimes described as the good cholesterol because it's removing peripheral cholesterol from the body. And LDLs are sometimes described as the bad cholesterol because they deliver cholesterol to the peripheral parts of the body. The reality is cholesterol is essential to cell function and cell structure. So they're not really bad or good. They're just functional from that perspective. Now, if we have excessive numbers of LDLs, it can indicate pathology. It can indicate delivery of too much cholesterol to the periphery. And that might increase the risk for atherosclerosis. And you'll learn about atherosclerosis when you talk about pathologies, but it's one of the leading factors associated with heart attacks and strokes. So we don't want too much cholesterol out in the circulation, but we do have HDLs that help us with that. Now, we'll talk a little bit more about those when we look at some of that metabolism that we have to talk about starting next week. So anyway, what we're talking about when we look at these hepatic sinusoids are that the result of collecting up that nutrient-rich venous blood is going to be visualized in that central region. In that last slide we looked at, which was where back on when we looked at this central canal, venous-rich, nutrient-rich venous blood with the sinusoidal capillaries and the cells lined up all around them makes for easy absorption of those nutrients. So our monosaccharides and our fatty acids and our amino acids are going to be readily distributed to the hepatocytes. So the hepatocytes can perform their metabolic, secretory, and endocrine functions. They've got the building blocks. They got first option, first pass at this location. We okay with that? You following me okay?
Yeah.
Sorta. We're gonna build on that. We're gonna come back to that when we look at metabolism. So it'll probably start to fall into play a little bit better there. So that vein, the hepatic portal vein, we've seen a portal vein before. We see there's a portal circulation between the hypothalamus and the anterior pituitary gland as well. But when you put that word portal in there, what that's saying is that you've got a capillary network that was originated from a set of veins. And so veins contribute to the entry point and the exit point. Normally, capillary beds are formed from an arteriole in and a venule out. In the case of a portal system, it's venule in and venule out. So we got the veins from the digestive system entering the veins of the kidney system, the liver system. That creates a hepatic portal system or a hepatic portal vein. So that's just terminology. That's where that came from. The other thing before we leave the anatomy of the liver is that there are a group of cells that are not hepatocytes. They're sometimes as a group called reticuloendothelial cells because they're associated with the endothelium, either just inside or just outside. But one of those cell types is called a Kupffer cell. And unfortunately, that name sort of sticks as far as that goes. But these are basically phagocytes. So they patrol the duct system and they patrol between the hepatocytes, and they're picking up debris. They're going to engulf and destroy worn out white blood cells, worn out red blood cells. They can release, for example, we know when we talked about red blood cells, that sometimes with red blood cells, we break down the hemoglobin and release the amino acids for resynthesis. Well, we're in the right location for that. We're in the liver, where these hepatocytes can pick up those amino acids and recycle them, rebuild them into something functional, maybe new hemoglobin. But the point is that the iron and the amino acids can be recycled, and the heme will get released into the circulation and contribute to the formation. It often is going to end up being part of bile, along with something called bilirubin, also a byproduct of breakdown of red blood cells. So they're macrophages. They're going to clean up. If there's bacteria, again, coming from the digestive system, there's a high possibility of that. These phagocytes can deal with that. And another cell type to be aware of is called a hepatic stellate. So that's going to be kind of a star-shaped cell. That's why it's called a stellate cell, found in the hepatic tissue. And most of the time, what we know about a stellate cell is most of the time it stays relatively dormant, not doing very much unless there's injury, either from infection or trauma, and then those stellate cells become active. Now, the issue with them is that when they become active, they create scar tissue. They don't encourage, for example, mitosis of the hepatocytes. They instead replace the damaged tissue with scar tissue. So these will become active under chronic exposure to toxins or injury, and they create scar tissue. So that represents a loss of function. So ideally, we don't want to activate those stellate cells for that particular purpose or reason. So just another cell type to be aware of when we look at what's going on in the liver. Okay, so let's look at some of these functions here. Liver. We mentioned that hepatocytes we know hepatocytes are going to produce bile. Almost all hepatocytes, amongst other factors and functions, produce bile salts. And those bile salts are going to become part of bile. And we know that that responsibility is for the the purpose of that bile is to emulsify fat. That's a type of mechanical breakdown that encourages more effective chemical breakdown. We also know that liver functions involve carbohydrate homeostasis. And that was what I used as our example. Carbohydrate homeostasis means that these cells can perform gluconeogenesis. Anybody remember what that meant? Is anybody still out there?
Yeah.
Gluconeogenesis. What are we talking about when we talk about gluconeogenesis?
Making new things from glucose.
Yeah, we're making new glucose from something other than a carbohydrate. So some of our amino acids are described as being gluconeogenic. Some of our fatty acids are gluconeogenic, and they can undergo that process as part of the functions of these hepatocytes. So hepatocytes are the cells that are capable of driving these equations. So glycogenesis is pretty straightforward. That's the formation of glycogen. That would be a step that would not affect, or at least would not increase blood glucose levels because we're making glycogen. But the other two mechanisms, gluconeogenesis and glycogenolysis, would both release glucose into the circulation and therefore raise blood glucose levels. And the other thing, so those all represent effects on blood glucose, carbohydrate homeostasis, but also the conversion of glucose to triglycerides, as we mentioned before. Then the triglycerides are going to be stored in adipose tissue. So these hepatocytes have at least these two functions. We also are going to find out that they're directly involved with lipid homeostasis. And by now we should be figuring that we're going to see protein homeostasis shortly, because that's the reason for this first pass, this first exposure of these nutrients to the hepatocytes, so they can determine the first pathway for these free fatty acids or monoglycerides or monosaccharides or proteins, amino acids, where they're going to go first. So we know that in lipid homeostasis, we can promote the storage of triglycerides. We can break down fatty acids to make those fatty acids available for ATP production. And we can produce those carrier molecules that I talked about on that other slide, produce LDLs and HDLs. So we deliver cholesterol to cells, and we remove cholesterol from cells. And the idea is that we're delivering the cholesterol that was produced in the liver to the cells, and we're picking up the cholesterol from the cells and bringing it back to the liver for either breakdown or reuse in the liver. So that's some of what's going on there, and we actually produce cholesterol. And if the lipids are required, they'll be released into the circulation, delivered to all the cells, so the cells can pick up the fatty acids and utilize them either to repair or restore structural or functional components within their responsibilities. So we know what's coming next: protein homeostasis. And in protein homeostasis, what we're talking about is these hepatocytes, they can remove that amine group. Remember, the amine group on an amino acid was the NH2 group? So they can remove that amine group from the amino acid, and that then allows that amino acid to be either converted to glucose, gluconeogenesis, or converted to pyruvate, or converted to acetyl coenzyme A. In either of those second two cases, pyruvate or acetyl coenzyme A, they're going to be available as the substrate that we need to make ATP in the Krebs cycle. Right? Does that make sense?
Yeah.
Now, the other thing that they do with respect to proteins is that they are going to be able to convert, well, they help to decrease toxicity. So sometimes when NH2 is removed from the amino group, it's going to pick up a hydrogen ion and become ammonia. And ammonia is potentially toxic. So what happens before the ammonia is allowed to accumulate is these hepatocytes can convert that ammonia to urea, which is much less toxic and is going to be excreted through the urine when it starts to accumulate. The other thing that the hepatocytes are capable of is protein synthesis. And in particular, we're talking about plasma proteins. Remember, the biggest contribution to plasma proteins was albumin. It's being produced here. But we also had plasma proteins that were released and only active under certain conditions, like fibrinogen, angiotensinogen, prothrombin, all examples of proteins that are being made in the liver and contribute to plasma protein, in other words, colloid osmotic pressure, unless they get signaled to do something else. Prothrombin and fibrinogen for clotting, and angiotensinogen for the conversion to angiotensin 1 and then angiotensin 2. So those are examples, other reasons for why the amino acids are going to pass first to the liver. Okay, now, so that we're up to 1, 2, 3, and 4. So number 5 is going to be the detoxification of some drugs and toxins. And those drugs include prescription type drugs, so-called legal drugs, and so-called street drugs. But the idea here is that the hepatocytes will recognize some of those potential toxins and be able to neutralize them. They can eliminate them or neutralize them by breaking them down and delivering them to the bile, and the bile will then be excreted through the digestive tract and so on. So that's what we're talking about when we detoxify drugs, toxins, and in some cases, certain hormones are going to end up being degraded down here at the liver as well. We can excrete bilirubin, and bilirubin comes from the heme that was part of hemoglobin in old red blood cells. So this bilirubin will be secreted into the bile. That's really where the name came from, and eliminated in the feces. So we'll say via feces, so we know it ends up in the digestive pathway. The other thing that these cells are able to do in the liver is the storage of vitamins and some minerals. So in particular, fat-soluble vitamins can be stored here. So those are A and E and D and K. The water-soluble vitamins are, by definition, water-soluble, can't be stored, so they're just going to be urinated out. They'll be filtered out through the blood in the kidneys and eliminated. So it's very difficult to get toxic with water-soluble vitamins. But because fat-soluble vitamins can be stored, there is the possibility of toxicity, overdosing of these vitamins, and that can become a problem. Now, in many cases, it's difficult to do that, but it is a possibility and something to be aware of if you're taking vitamin supplements with respect to the fat-soluble vitamin supplements. You need to be aware that they can accumulate in tissues, particularly these tissues, the hepatocytes. So some of the minerals that are stored are iron and copper and zinc are some examples of some of the minerals that are stored in these cells. And we've talked a little bit about these. These minerals are nutrients for some bacteria. And what happens in fever is it encourages these hepatocytes to keep the iron, copper, and zinc, or reuptake iron, copper, and zinc. And that takes away a potential source of nutrients for some of these bacteria that may have been causing the fever in the first place. So fever encourages the reuptake of these types of minerals into the hepatocytes and becomes a way of helping to control an infection by taking away a food source for what was potentially the cause. And activation of pre-vitamin D. Remember way back when we talked about the very first system in AMP 100, the skin, and keratinocytes produced pre-vitamin D when the keratinocytes were exposed to sunshine. And pre-vitamin D was not yet active, thus pre. And so it needs to be converted both at the kidneys and at the liver in order to become active. So this becomes an important role for these hepatocytes. And it would actually be the addition of an OH group to the pre-vitamin D molecule that is part of the activation process. And we have to do that 2 times, once at the liver and once at the kidneys. So it plays a role in the activation of pre-vitamin D. Okay, so the idea of spending a little extra time on the liver is to really appreciate the additional functions and responsibilities that the liver has. Okay, so we need to be very aware of that. Now, to sort of summarize up what we've been talking about, we can describe phases of digestion. Phases of digestion can be described as cephalic. So cephalo means towards the head. You'll sometimes see it described as psychic. So that gives you a clue too. But cephalic phase of digestion is what happens when you think about food, when you smell food, and it will literally initiate the production of gastric secretions. So, so that's the thought, sight, smell of food. So that's literally not physical. It's psychological, it's cephalic referring to the head. It's what happens when you start to think about food, either consciously or subconsciously. Then there's a gastric phase. So that's what happens when food actually enters the stomach. So food, especially amino acids, gastric phase. So that's physically going to cause a stretch, and stretch is going to be monitored by receptors that will signal the vagus nerve to release ACH. And we already know what's going to happen when the vagus nerve releases ACH. It's going to stimulate the parietal cells to produce hydrochloric acid. So that's where the secretions are going to come from. So we get stretch in the stomach and the presence of food, especially, is going to promote the production and release of gastrin. Now, then we get the intestinal phase. So we've got a cephalic phase, a gastric phase, and now we're down into the intestinal phase. And at the intestinal phase, this is what's happening when the duodenum responds to chyme and releases hormones to promote digestion and absorption. So we're going to talk a little bit about those hormones that are going to be involved to sum up what we've been spending so much time on today. So when we look at endocrine factors, we've got a hormone called ghrelin. Ghrelin is produced by a cell that's called the ghrelin cell, for lack of a better name, that is also a cell found in the stomach. We didn't mention it at the time, but produced by gastric cells. We're just going to call them ghrelin cells, so we don't get them mixed up with G cells or any of the other cells that we already talked about. But these cells are going to get excited when the stomach's empty. So what happens is they send signals to the hypothalamus to stimulate the hunger centers in the hypothalamus, and also signal the anterior pituitary gland to release growth hormone. So sometimes it's called a growth hormone secretagogue, because a secretagogue is a synergistic signal to another hormone. If it happens to be named as a growth hormone secretagogue, it's synergistic for growth hormone. So released when stomach is empty, to signal hypothalamic hunger center and the anterior pituitary gland to release growth hormone. Now, leptin is sometimes described as the antagonist of ghrelin. Leptin is released from adipocytes and it signals nerves and hypothalamus to decrease hunger. So the fancy word for when you're full or when your metabolic needs have been met is you're sated. Satiation is the opposite of hunger. So when sated means that your glucose levels are adequate, your lipid levels are adequate, and your protein levels are adequate. So in other words, it's signaled to decrease hunger. Okay, and we know that what happens in obesity, or at least in a percentage of people with obesity, they have abnormal responses from leptin. In other words, they don't receive the decreased hunger signal from the hypothalamus as efficiently or effectively as people who don't have that particular type of obesity problem. Now, a couple other hormones. We mentioned gastrin. We already know that gastrin is released from G cells and stimulates HCl production from our parietal cells. Okay, it also relaxes the pyloric sphincter. So if we relax the pyloric sphincter, what's the significance of that? The pyloric sphincter was a sphincter that emptied into the duodenum. So if we relax it, what happens?
Get acid into the stomach.
Yeah, so this is one of the ways we're going to deliver chyme from the stomach into the small intestine. Makes sense that if we're producing gastric secretions and we're initiating both that mechanical and that chemical digestion in that process, that we're going to want a place to go to once we've done that. So gastrin levels help relax that sphincter. Okay, I mentioned that hormone called cholecystokinin, TCK, cholecystokinin, and cholecystokinin inhibits gastric emptying. So there's our understanding that gastrin would promote gastric emptying and cholecystokinin inhibits it. So those two are going to work as homeostasis on stomach emptying. So we also know, because we talked about it already, that it stimulates contraction of the gallbladder so that we're going to deliver bile. It stimulates pancreatic secretions. So it gets released when food is introduced to the duodenum. So that makes sense, right? We don't want too much food being introduced to the duodenum. So we're going to inhibit gastric emptying. We're going to stimulate the gallbladder and stimulate the pancreas, though, to neutralize the contents from the stomach and to start working on emulsification of lipids. So that's what cholecystokinin is going to be doing. Okay, and as a result, a result of those things, we're also going to neutralize some of that pH. All things that we want to see happen here. Now, secretin is also produced by cells in the small intestine and has overlapping functions with cholecystokinin. It's going to stimulate pancreas, So I'm just going to try to abbreviate it here because we're running out of space. Stimulate the pancreas, that helps to neutralize, stimulates bile production, and inhibits gastric emptying. So a lot of the same features that cholecystokinin is responsible for, secretin is also responsible for. So secretin promotes secretion. That's essentially what we're talking about there. And stimulates. So it's a little bit different here. Bile production, not so much a direct contact onto the gallbladder, but more about stimulating bile production from the liver. So more bile will be produced under the influence of secretin. So these are just some examples of the endocrine influences on the digestive process. Okay. So of all those secretions that are being produced, from saliva, from gastric, from intestinal, from the pancreas and the bile, represents thousands of milliliters per day. And we reabsorb most of that so that we only lose about 200 milliliters per day through the stool. So we might be producing somewhere in the vicinity of, 8 or 9,000 milliliters, 8 or 9 liters of fluid per day on those secretions. And we only need to replace about 200 milliliters of that in the interest of homeostasis, because the rest of it's reabsorbed along the way. Okay, I think that's enough about the digestive system. Like I said, we got test number 3 next week.